Okay, today I'm going to go through what's called the central dogma of biology. It's called gene expression, is another term for it. So with this part of the class, we've learned about DNA structure, we've learned a little bit about proteins, now we're going to kind of merge the two together. So in the central dogma, there's kind of two steps to it, and there's three parts to that two-step process. The first is starting with DNA, which is called the gene part of it. There's an intermediate, which is called RNA, which is a messenger. And then there's the amino acids and the proteins that are the expression of the gene. And so where this occurs in the cells is, you'll see here that here's a cell with a nucleus. And in the nucleus is the DNA strands. Now the DNA strands cannot leave the nucleus, but that's where the RNA messenger comes in. So the RNA can take the message from the DNA, leave the nucleus, and then produce the proteins from that. And sometimes those proteins get exported to other cells in the body, as I'll show you here. So, just a few things about proteins. They are specific to certain cells. So some cells produce one protein more than others. So here's 4 examples that I have. The protein called actin is in the cell types muscle and heart cells, and it's used for contraction or movement. So this specific protein is used in those cells more often because they do more contraction and more movement type of functions. Insulin is a protein that's produced in the pancreas. This is used for sugar digestion. Lipase is an enzyme, that's produced in the small intestine. So enzymes are a part of proteins. Lipase is produced in the small intestine, and it's used for fat digestion. Similar to the word lipid, which we learned earlier. Melanin, which we talked quite a bit about, is in hair, skin, and eyes, and that gives the brown color that we see in people who have darker skin, darker eyes, or darker hair. And the main function of it really is to protect those people from sunlight. So a little bit about protein structure and how that works. The protein is structured into amino acid parts. So amino acid 1, amino acid 2, 3, 4, 5, 6, and oftentimes amino acid chains will be at least 20 for a protein, and they can be up to 500 or more amino acids in length. And so some of the shapes that come out of proteins are like sort of like a spiraling. We call it an alpha helix. So you have to imagine this might be a chain of 100 amino acids that are linked into a spiral shape. Another type could be what's called a beta sheet, which kind of goes back and forth into a flat surface. And so these are the two main shapes that we see, aside from just kind of random shapes within the protein. So this is what a protein might look like, where it might have 2 different alpha helixes and 3 different beta sheets. And then the purple region just shows kind of what's called the primary structure, where it's just kind of in a random shape. Now, these are specific arrangements, so the protein doesn't just assemble randomly. So, for example, insulin, which is used for sugar digestion, will always have the same shape to it because it always has the same function. So protein function and shape are directly related to each other. So, how do we get to proteins? There's 2 parts to it. The first part's called transcription. This occurs in the nucleus. It's the process of going from DNA to messenger RNA. So let's take the DNA strand. Now this is a very small piece of DNA, just so I could fit it on the board. And the activities we do in class will also be smaller pieces, but understand that this DNA could be hundreds of letters long. So I've done one that is 12. And it's typical to take and break the DNA into groups of 3. And you'll see that later on as I do the second part of this. But basically what happens in the nucleus, the DNA gets converted to RNA. And it gets converted to its complementary letter. So, for example, if the DNA is a T, the RNA is going to be an A. If the DNA is an A, the RNA is going to be a U, because remember, we don't have T's in RNA. Usually that would be a T. Since it's RNA, it's a U. The next letter is a C, so we're going to have a G here. We have a G here, so we're going to have a C, G, C, A, U, C, G, T, A, A, U, and then your UAA to finish off there. So this is our RNA strand that's then going to leave the nucleus, and it's going to travel to one of 2 spots, either the ribosomes or the rough ER for the second part of producing a protein, which is called translation. Translation is taking the RNA, converting it to the protein. So taking the messenger RNA. So the messenger RNA strand from the previous board, AUG, CCU, GAU, and UAA, will produce the amino acid sequence, and this comes from the chart that's in your review guide, or I handed it out in class, one of the two. So this amino acid chart has 3 letters combinations for each of the amino acids, and those are called codons. So they usually start with the AUG because that is the start codon for many proteins. So we usually look for that first. Where's that at? Here it is. So it's called methionine is the name of the amino acid, but it's also the starting one. CCU, if you look that on your chart, you'll find that that's called proline. GAU, if you look at that, it's called aspartic acid. And then the fourth one, which I have here, UAA, is actually a codon for stop, which means the protein would stop being built at that point. And so this protein is only 4 amino acids long. Like I said, that's very short. There's no amino or no proteins that short. The shortest ones are in the range of 20 amino acids. So, kind of a review here. Talk about the central dogma, the process of taking DNA, changing it to RNA, changing that to proteins through transcription and translation. So these are the 2 processes you need to know how to do, and we'll practice. This can be related to what's called gene expression. So taking a gene of DNA, using a messenger to convert that to the gene expression. What does it look like in the person? And then here's an example. So the insulin gene is 165 letters or nucleotide bases long. Insulin, the amino acid, the protein, is 55 amino acids in length. So that's where the 165 bases comes from, because it's a multiple of 3. So that's it for the central dogma and gene expression. Make sure you go on and take the review quiz.